Welcome to this bioprocess engineering online tutorial on animal cell kinetics in bioreactors. In this tutorial, we will explore how animal cells grow and behave in bioreactors and how their kinetics can be described and modeled for bioprocess design and optimization. In the Bioprocess Engineering online tutorial series, I present the fundamental principles of bioprocess engineering and their industrial applications in microbial fermentation, enzymatic bioconversion, and animal cell culture processes. For more tutorials and updates, please subscribe to our YouTube channel or visit our website. In this tutorial, you will learn the fundamentals of animal cell kinetics in bioreactors, including cell growth, substrate consumption, and metabolite production. By the end of this tutorial, you will be able to answer the following questions. Why is cellular kinetics important in bioprocess engineering? What are the main kinetic rates used to describe animal cell cultures? Which kinetic models are most commonly used to describe cell growth, substrate consumption, and metabolite production? Let's begin with a fundamental question. Why is it important to study animal cell kinetics? Animal or mammalian cells play an increasingly important role in the biopharmaceutical industry. Their unique ability to perform complex post-translational modifications makes them the preferred platform for producing many high-value therapeutic products. Today, animal cell cultures are widely used to manufacture recombinant glycosylated proteins, especially monoclonal antibodies, viruses for vaccine production and gene therapy, and even living cells themselves, which serve as the final product in cell-based therapies. To manufacture these biopharmaceuticals, animal cells are cultivated in a wide variety of bioreactors. At laboratory scale, roller bottles and spinner flasks are commonly used for process development. At industrial scale, production is typically carried out in stirred tank bioreactors, which provide the precise environmental control required for large-scale cell culture. Animal cells can be cultivated in bioreactors using two main growth modes. The first is suspension culture, where cells grow freely in the liquid medium. A well-known example is Chinese hamster ovary, or CHO cells, which are the industry standard for producing recombinant glycoproteins, including monoclonal antibodies. The second is adherent culture, where cells grow attached to a solid surface, such as microcarriers suspended in the bioreactor. Examples include Vero cells, derived from monkey kidney epithelial cells and widely used for viral vaccine production, and HEK cells, or human embryonic kidney cells, which are commonly employed to produce therapeutic proteins and viral vectors for gene therapy. During a cell culture process, several key variables can be measured experimentally. These include the consumption of essential nutrients such as glucose, glutamine, and oxygen. We also monitor the cell population by measuring the concentrations of viable and dead cells. In addition, we quantify metabolic byproducts such as lactate, ammonia, and carbon dioxide. Finally, we measure the production of the target bioproduct, which may be a recombinant protein, a virus, or another therapeutic molecule. Together, these measurements provide a comprehensive picture of the culture's physiological state and performance. Cellular kinetics is the study of the rates at which cellular processes occur. Its objectives are threefold. First, to quantify important cellular rates, including cell growth and death, nutrient consumption, metabolite production, and, when applicable, the formation of recombinant proteins or viruses. Second, to identify the factors that influence these rates, particularly the composition of the culture medium and the surrounding environment. And third, to develop mathematical models that describe these rates as functions of the relevant biological and environmental variables. These kinetic models form the foundation of quantitative bioprocess engineering. Cellular kinetic models are a key component of mechanistic simulation models for animal cell cultures. When combined with other fundamental phenomena, such as mass transfer, mixing, and fluid flow, they allow us to simulate the dynamic evolution of the bioreactor. These models can predict how cell density, nutrient concentrations, metabolite levels, and product concentration change over time. Simulation models that incorporate cellular kinetics can be viewed as digital representations or digital twins of real bioreactors. They enable engineers to evaluate operating conditions, optimize process performance, and investigate scale-up strategies before carrying out expensive laboratory or pilot-scale experiments. As a result, they reduce development time, lower costs, and accelerate process optimization. Another major advantage of these simulation models is their ability to predict culture performance under different operating modes. They can describe the time evolution of cell growth, substrate consumption, metabolite production, and product formation in batch, fed batch, and continuous cultures. Understanding these dynamic behaviors is essential for improving process productivity, maximizing product yield, and ensuring robust and consistent industrial manufacturing. In the second part of this tutorial, we will introduce the concepts of cellular specific rates and kinetic laws. These are the fundamental tools used to describe and model the behavior of animal cells during culture. Animal cell kinetics is described using specific rates, which are expressed per million cells and per hour. First, there are the specific substrate consumption rates, expressed in moles consumed per million cells per hour. Examples include QG-LC for glucose, QGLN for glutamine, and QO2 for oxygen. Next, we have the specific cell growth rate, mu, and the specific cell death rate, KD, both expressed in reciprocal hours. We also define the specific metabolite production rates, expressed in moles produced per 10 to the 6 cells per hour. Typical examples are QLAC for lactate, QNH4 for ammonia, and QCO2 for carbon dioxide. Finally, the specific protein production rate, QProt, represents the amount of recombinant protein produced per million cells per hour. Together, these specific rates provide a quantitative description of cell metabolism, growth, and productivity. The specific rates of animal cells are not constant. They depend on many environmental conditions, which usually change throughout the culture. The first group of factors is the availability of nutrients in the culture medium. These include glucose, glutamine, other amino acids, peptides, growth factors, and dissolved oxygen. The second group is the accumulation of metabolic byproducts, particularly lactate, ammonia, and, in some cases, dissolved carbon dioxide. As these compounds build up, they can inhibit cell growth and productivity. Specific rates are also influenced by physicochemical conditions, such as temperature, pH, and osmolarity. Finally, cells may experience physical stresses, for example, those generated by agitation in the bioreactor, or by the formation and bursting of air bubbles. All of these factors interact to determine the physiological state of the cells, and consequently, their kinetic behavior. The influence of environmental conditions on cellular behavior is described by kinetic laws. These are mathematical relationships that predict specific cellular rates from variables such as nutrient concentrations, metabolite concentrations, temperature, or other culture conditions. In animal cell kinetics, two main categories of kinetic laws are commonly used. The first category is rate-environment relationships. These express a specific cellular rate, such as the cell growth rate or the cell death rate, as a function of one or more environmental variables. The second category is rate-rate relationships. These relate one specific rate to another. For example, the substrate consumption rate may be expressed as a function of the cell growth rate, and the metabolite production rate may be related to the substrate consumption rate. Let us now examine the kinetic laws that describe cellular growth in more detail. We will see how the different phases of cell growth can be represented mathematically, and how environmental conditions influence the specific growth rate. Whether they grow in suspension or attached to a surface, animal cells generally follow the same sequence of growth phases in a bioreactor. The first stage is the lag phase, during which cells adapt to their new culture environment. Cell proliferation is still limited during this period. This is followed by the exponential growth phase, where cells divide rapidly and the cell concentration increases almost exponentially. As nutrients become depleted and metabolic byproducts accumulate, the culture enters the reduced growth phase. Cell proliferation gradually slows down. Finally, the culture reaches the decline phase, during which the number of viable cells decreases because cell death becomes dominant. Understanding these different phases is essential for modeling animal cell cultures. The lag phase corresponds to the period during which freshly inoculated cells adapt to their new environment. During this stage, the cells are assumed to be initially in a latent or dormant state with very little metabolic or proliferative activity. As adaptation proceeds, these latent cells gradually become actively growing viable cells. The transition rate, denoted R-lat, is expressed in millions of cells per liter per hour. A common assumption is that this transition follows a first-order kinetic law, meaning that the transition rate is proportional to the concentration of latent cells. The proportionality constant, K-lat, is called the transition rate constant. Its value determines the duration of the lag phase. The larger K-lat is, the faster the cells adapt and the shorter the lag phase. Once the cells have adapted to the culture conditions, they enter the exponential growth phase. During this period, cells divide at their maximum specific growth rate, denoted mu-max. For a batch culture, the cell balance leads to the differential equation, dx over dt equals mu max times x. Integrating this equation gives the classical exponential growth expression, x equals x0 multiplied by the exponential of mu max times t, where x0 is the viable cell concentration at the beginning of the exponential phase. For most industrial mammalian cell lines, the maximum specific growth rate typically ranges from 0.02 to 0.04 per hour, corresponding to a doubling time of approximately 1 to 2 days. As the culture progresses, nutrient concentrations decrease and cell growth gradually slows down. The most widely used model describing this effect is the Monod equation. According to this relationship, the specific growth rate equals the maximum growth rate multiplied by a substrate limitation term. Mu equals mu max, multiplied by S divided by Ks plus S. Here S is the substrate concentration and Ks is the half saturation constant. It represents the substrate concentration at which the growth rate reaches half of its maximum value. In mammalian cell cultures, glucose and glutamine are the nutrients that most frequently become limiting. The Monod equation is therefore often extended by combining two limitation terms, one describing glucose limitation and the other glutamine limitation. In this way, the growth rate decreases whenever either nutrient becomes insufficient. For adherent cells, growth depends not only on nutrients, but also on the amount of available surface. As cells progressively cover the culture surface, or the microcarriers, less free space remains for new cells to attach and divide. This effect is described by introducing the maximum cell concentration at confluence, denoted X max. The specific growth rate is then expressed as the maximum growth rate multiplied by the term 1 minus X divided by X max, raised to the power alpha. The exponent alpha, which is greater than 1, controls how sharply the growth rate decreases as confluence is approached. The graph illustrates this behavior for 3 values of alpha, 1, 2, and 5. As the value of alpha increases, the growth rate remains close to its maximum for longer, then decreases more abruptly near confluence. Growth can also slow down because of the accumulation of metabolites released by the cells. The two most important inhibitory metabolites are lactate and ammonia. A commonly used inhibition model expresses the specific growth rate as mu equals mu max, multiplied by 1 minus m divided by m max, raised to the power alpha. Here m is the metabolite concentration, m max is the concentration at which growth completely stops, and alpha is an empirical exponent, usually greater than or equal to 1. The graph shows how the normalized growth rate varies with the normalized metabolite concentration for different values of alpha. When alpha equals 1, the inhibition is linear. For larger values of alpha, the inhibitory effect is initially weak, but becomes much stronger as the metabolite concentration approaches its critical value. In practice, mammalian cell growth is usually inhibited by both lactate and ammonia. The complete kinetic expression therefore includes two independent inhibition terms, one for each metabolite. The final decline phase is characterized by a decrease in viable cell concentration as cell death progressively exceeds cell growth. One of the main causes of this increase in cell death is the accumulation of toxic metabolites, particularly lactate and ammonia. A simple model assumes that the specific death rate increases exponentially with the metabolite concentration. Kd equals Kd0, multiplied by the exponential of m divided by Kdm. Here, Kd0 is the minimum specific death rate observed under favorable culture conditions, while Kdm is the death rate acceleration constant. When the metabolite concentration reaches Kdm, the specific death rate is multiplied by approximately 2.7. Since both lactate and ammonia contribute to cell death, the complete kinetic model generally includes two independent exponential acceleration terms, one for each metabolite. Let us now examine the kinetic laws that describe substrate consumption rates, focusing on the major nutrients, glucose, glutamine, and oxygen, required for animal cell growth and metabolism. During cell culture, the rate at which cells consume nutrients is closely related to their growth rate. The specific substrate consumption rate, denoted QS, is expressed in moles of substrate consumed per million viable cells per hour. A widely used kinetic expression is QS equals YS over X multiplied by mu plus MS. This equation contains two additive contributions. The first is the growth-associated consumption, which is directly proportional to the specific growth rate. The coefficient YS over X is the stoichiometric yield, representing the amount of substrate required to produce 1 million new cells. The second contribution is the maintenance consumption, represented by MS. This term accounts for the nutrients consumed to sustain basic cellular functions, even when the cells are no longer dividing. At high growth rates, substrate consumption is dominated by the growth-associated term, while the maintenance contribution remains relatively small. This kinetic law can be applied to all major substrates, including glucose, glutamine, and oxygen, each with its own stoichiometric yield coefficient and maintenance constant. In cultures of recombinant cells that secrete large quantities of protein, the substrate consumption rate law includes an additional term associated with recombinant protein production. The commonly used kinetic expression is QS equals YS over X times mu plus MS plus YS over P times QP. The first term represents growth-associated substrate consumption required for biomass formation. The second term represents substrate consumption for cellular maintenance. The third term represents protein production-associated consumption. It is directly proportional to the specific protein production rate, QP. The coefficient YS over P is a stoichiometric coefficient expressing the amount of substrate consumed per unit mass of recombinant protein secreted, for example in moles of substrate per gram of protein. In the final part of this tutorial, we will examine the kinetic laws that describe the production of metabolites and recombinant proteins during animal cell culture. The production of metabolites excreted during cell growth, such as lactate and ammonia, is closely linked to substrate consumption. The specific metabolite production rate, denoted QM, is expressed in moles of metabolite produced per million viable cells per hour. It is commonly related to the substrate consumption rate by the simple stoichiometric relationship. Qm equals Ym over S multiplied by Qs. The coefficient Ym over S represents the stoichiometric yield of metabolite formation expressed as moles of metabolite produced per mole of substrate consumed. This relationship applies to the principal metabolites encountered in animal cell culture. Lactate is mainly produced from glucose metabolism. Ammonia originates primarily from glutamine metabolism, and carbon dioxide is generated during the oxidative metabolism of substrates through cellular respiration. Each metabolite is characterized by its own stoichiometric yield coefficient, which depends on the metabolic pathways active in the cells. For some metabolites, the production yield is not constant. This is particularly true during long-term fed batch or continuous cultures, where glucose is intentionally maintained at a low concentration. Under these conditions, cells progressively adapt their metabolism and produce significantly less lactate. As a result, the glucose to lactate yield is no longer constant. At high glucose concentrations, the yield reaches its maximum value. As glucose becomes limiting, the yield decreases. The dependence of Y-lac over GLC on glucose concentration can be described by a sigmoid-shaped glucose limitation term, characterized by the parameter K-lac over GLC. Y-lac over GLC equals Y-lac over GLC max, GLC to the power n, divided by K-lac over GLC, plus GLC to the power n, where n greater than 1 determines the steepness of the transition between low and high-yield regions. This model captures the well-known metabolic shift toward a more efficient use of glucose under glucose-limited conditions. A similar phenomenon is observed for ammonia production. Ammonia is generated during glutamine metabolism. When cells are maintained for extended periods under low glutamine concentrations, they gradually reduce ammonia production. Consequently, the glutamine to ammonia yield decreases as glutamine becomes limiting. As for lactate production, this variation can be represented by a sigmoidal kinetic law. The maximum yield is obtained at high glutamine concentrations, while the yield progressively decreases under glutamine limitation. This model reflects the metabolic adaptation of animal cells to nutrient-limited environments and improves the prediction of ammonia accumulation during long-term cultures. For recombinant cell lines, modeling protein production is generally more complex than modeling metabolite production. Unlike substrate consumption or metabolite formation, recombinant protein production is often not directly associated with cell growth. During the initial exponential growth phase, cells primarily invest their resources in proliferation, and protein production remains relatively low. The highest production rates are frequently observed later in the culture, when cell growth slows down or even stops. The specific protein production rate depends on several factors. It is influenced by the availability of substrates such as glucose and glutamine. It is reduced by the accumulation of inhibitory metabolites, particularly lactate and ammonia. It also depends on the cellular growth rate itself. Finally, high concentrations of the secreted recombinant protein may reduce further protein production through product inhibition. To account for all these effects, the maximum specific protein production rate is multiplied by several kinetic terms describing substrate limitation, metabolite inhibition, product inhibition, and growth rate inhibition. The exact mathematical expression and the associated kinetic parameters depend on the cell line and the recombinant protein being produced. In summary, what have you learned in this tutorial? In this tutorial on animal cell kinetics, you have explored the main kinetic laws used by bioprocess engineers to describe and predict the behavior of animal cell cultures in bioreactors. These models make it possible to simulate how cell concentration, substrate concentrations, metabolite concentrations, and recombinant protein production evolve throughout a culture. More specifically, you have learned about specific growth rate models, including the effects of nutrient limitation, surface limitation for adherent cells, and inhibition by metabolites. Specific cell death models, describing the acceleration of cell death as toxic metabolites accumulate. Specific substrate consumption models, which distinguish between nutrient consumption required for cell growth and that required for cellular maintenance. metabolite production models, which relate metabolite formation to substrate consumption while accounting for metabolic adaptations under nutrient-limited conditions. And finally, recombinant protein production models, which describe how protein synthesis depends on nutrient availability, metabolite accumulation, cell growth, and product inhibition. Together, these kinetic laws form the foundation of mathematical models used to simulate, optimize, and scale up animal cell culture processes. If interested, you can gain a deeper insight in animal cells kinetics by enrolling to the online courses Introduction to Animal Cell Culture Technology and Engineering, and Digital Scale-Up and Optimization of Animal Cell Culture Bioreactors. For further information, please visit our Bioprocess Engineering online website or contact us at bioprocessdigital.com. We hope you found value in this introductory tutorial on bioprocesses. Your feedback is important to us, so please share your comments with us. Explore our website for more information about our comprehensive online courses in bioprocess engineering, and be sure to subscribe to our Bioprocess Engineering online channel for upcoming tutorials. Thank you for watching.